Hello everyone, and welcome to today's live broadcast, Impact of Next Generation Sequencing on Precision Medicine, presented by Gary Schroth, PhD, a distinguished scientist at Illumina, Inc. I am Kristen Perret-Gentile of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. And if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Now please join me in welcoming Dr. Gary Schroth. I will now turn the presentation over to him.
Thank you very much. It's great to be here, and I want to thank LabRoots for inviting me to give a presentation. So my name is Gary Schroth. I am a vice president in our product development group here at Illumina, and I've been working in the field of next generation sequencing for just over 12 years now. I was part of the company called Solexa that Illumina acquired 10 years ago, and which got Illumina into the field of next generation sequencing. And so this is actually kind of been a momentous year for us. We've celebrated our 10-year anniversary of the acquisition of Solexa by Illumina, and so it has reminded us. It's kind of fun to look back on the past 10 years. And so the first few slides I have talk about our technology and the 10 years of sequencing innovation that at Illumina we have driven in our product development group. And for those of you familiar with our platforms, you probably remember the Genome Analyzer, which we launched about 10 years ago, followed by the HiSeq 2000, which really a huge increase in output. And then the HiSeq 2000 was iterated into the 2500 and the HiSeq X10 system, which brought on board pattern flow cells. It was really the system that enabled the $1,000 genome in the 2014 timeframe. And during that time, we also were working on much smaller benchtop desktop instruments like the MiSeq, the NextSeq, and the MiniSeq. And you can see listed below all the types of technology breakthroughs that we have had over the years, over the last decade, that have enabled us to continue to create better, faster, cheaper systems in a way that we can drive down the cost of DNA sequencing significantly. That's shown on this plot here. So if you look at this plot, this is comparing the cost of genome sequencing an entire human genome, the cost of sequencing, and you can see that in about 2007 it was still several $1 million to sequence a genome. And when we launched the Genome Analyzer in 2007, 2008, we were able to bring that cost down to hundreds of thousands of dollars, which is a significant drop. Then when we launched the HiSeq 2000 system in around 2010, we brought it down into the tens of thousands of dollars, and we iterated the HiSeq 2000 system over the last 4, 5, 6, 7 years, and now basically on the HiSeq 2000 or the HiSeq X system, the X10 platform, you can actually get down to $1,000 genome prices. So this is several orders of magnitude drop in sequencing price over the last 10 years. And what that has led to is the world has sequenced a lot more genomes because of that, because it's a lot less expensive. So if we think back to 10 years ago, there literally were just 2 or 3 genomes that had ever been done. In the very first genome was Craig Venter's genome that was done using capillary electrophoresis in the early 2000s. The 454 platform generated Jim Watson's genome for about $2 million in 2007. There were several genomes done on the original GA and GA2, and you can see the price dropping as we moved into the 2000s in the HiSeq 2000 system. So you can see that it's down to about $10,000 genome, and by this point there were thousands of genomes done, but still not an enormous amount that we see today. By most estimates, we believe that in 2017 there will be upwards of 1 million genomes sequenced using our technology. And so another kind of interesting thing to look at is the throughput of our high throughput systems in terms of the amount of data that each system can generate. And I've made this this sort of diagram here showing the GA2 in 2008 generated about 4 gigabases of data. So that's 4 billion base pairs of sequence information basically in about a week it took to run that instrument to generate that much data. And that was a lot of data in the day. This was back in the day when genome centers wouldn't even generate that much data in a month from the entire center. And so this was a huge breakthrough in sequencing, and as we just talked about, the cost. And if you think about it in terms of hours per gigabase, it took 42 hours to generate 1 billion base pairs of sequence information, which, as I said, in 10 years ago, that was really, really impressive. Then when we launched the HiSeq 2000 system in 2010, you can see that the throughput went up significantly, and the runtime stayed about the same. So then it was about an hour per gigabase. And then what followed then was the HiSeq X system, where we had optimized the HiSeq system to generate much higher throughput and much less runtime. Now we're down to 2.4 minutes per gigabase. And then finally, what I'm going to talk about today in the first half of my talk is our latest system, which is called the NovaSeq, and I'm going to describe this system in detail. And this system is another sort of quantum leap in terms of how much data we can generate and also the runtime. And we've dropped the time per gigabase down to 40 seconds per gigabase. So this is what I'm going to talk about in the first half of the talk is the NovaSeq series. The second half of the talk, I'll talk about some specific applications related to precision medicine, especially cancer. So I want to introduce the NovaSeq series. We launched this system earlier this year. We have actually been shipping them starting in March. If you follow Illumina, you know that we've actually had quite a lot of interest in this instrument and quite a lot of orders, as we announced at our recent earnings call a couple of weeks ago. But I want to give you a little bit of technical background on this system. It's really a breakthrough system, very scalable platform, very flexible, very streamlined. And just like the HiSeq system took us from hundreds of thousands of dollars per genome down to the $1,000 genome, this system is starting at the sub $1,000 genome, and we think this platform and this system is going to be able to drive us down to the $100 genome in the next few years. So the NovaSeq system is actually sold in 2 different configurations. We call it the 5000 and the 6000. The only difference between these two is that the 5000 holds a single flow cell, the 6000 holds 2 flow cells. Also, in terms of the flow cell availability for each is slightly different. Obviously, the 5000 is a little bit less expensive. But the main thing I want to talk about today are the 4 types of flow cells that we have. Right now, we have launched with just this flow cell. If you look here in this corner, you can see the names of the flow cells up here: S1, S2, S3, and S4. Those that nomenclature is sort of our naming convention for these flow cells, and each flow cell has this sort of output: a half a terabase, so that's 500 gigabases, 1 terabase, 2 terabase, and 3 terabase. So that's 3,000 gigabases of data from each flow cell. So the NovaSeq 6000 system, for instance, can run 2 flow cells at a time, each one producing 3 terabases of data. And if we look at the output of this in a little bit more detail, you can see that we have over here on the left-hand side of the screen, you can see the flow cells types over here, and then you can see the sort of cycle configurations here. So if you look at the flow cell types S1 through S4 and the cycle configurations 100 cycle, 200 cycle, 300 cycle, you can see the range of outputs that we get here in the middle in terms of how many reads we can get in this column, or how many gigabases of data we can get here in this section. And so this allows you to go from 167 gigabases of data using a 100 cycle kit all the way up to 3,000 gigabases of data using the 300 cycle kit, and a range of reads from 1.6 billion up to 10 billion. So quite a lot of flexibility across these various flow cells. And so we'll talk about how we engineered that and how we built this system as we go through the talk. So I like to look at some of the enabling technology that's built into the system. And one of the key things about this system is that it is completely different than our HiSeq platform. It's a completely reimagined, re-engineered, sort of from the ground up new technology. And so the HiSeq 2000, 2500, 3000, 4000, and HiSeq X systems are essentially the state-of-the-art genome sequencers today. All the genomes that are going to be done last year and this year are mostly going to be done on the HiSeq X system that have been out there for a couple of years. Those systems generate about 16 genomes worth of data every 3 days or so. The NovaSeq is completely reimagined, where we've taken the HiSeq architecture, the imaging module, the fluidics modules that are built into the instrument and also into the CBOT system, and also the computer, and we've combined them all into one floor standing unit. Whereas the HiSeq sits on a bench, these instruments are floor standing. They include a very powerful compute engine, really nice integrated fluidics, and a very powerful imaging module. So the key technology enablers from this system, which as I mentioned is sort of redesigned from the ground up, we've designed completely new high density flow cells. We have, along with that, come up with an entirely new surface chemistry to increase the signal to noise, new superior very fast imaging. That's one of the keys to this system. And like I said, it includes a really quite powerful built-in computer that does a lot of data management, primary analysis. Our RTA, real-time analysis software, is 8 times faster than the previous instruments. And we've reformulated the chemistry that does our SBS chemistry, as we call it. So one of the key things to this new system is the new pattern flow cell. So if you look at our flow cells in the past for the HiSeq and the HiSeq X system, you can see they're relatively a lot smaller than the NovaSeq flow cells, and so there's a lot less surface area. And also we use this patterning technology where we put little wells into the surface of the flow cell, and you can see that we are packing the wells in slightly tighter and slightly smaller wells as well. And so if you look at kind of the amount of surface area we're looking at, plus the tighter pitch and smaller wells, we can actually put quite a bit higher density of information into the same sort of square millimeter, and then we have a lot more square millimeters to scan, so we could overall generate quite a lot of output. The thing about this system that is amazing is that it can scan this large area, which is obviously much larger than this, actually faster than we would scan this area in the older systems. So this shows the clustering chemistry. This system also does all onboard clustering. And if you've ever used our previous systems, you know some of the desktop ones do onboard clustering, but the HiSeq X and HiSeq systems use a CBOT, so we no longer need this. And we've also worked on the formulations and configurations of these protocols to make everything a lot faster than it was on the HiSeq X system. This is what we call a two-channel chemistry, two-channel SBS chemistry, and so this is an evolution of our chemistry that we started on the NextSeq several years ago, NextSeq version 2 chemistry. We actually use a very similar chemistry on the mini-seq system, and so this NovaSeq version of this chemistry is probably our V4, V5 of this sort of chemistry in our total portfolio. And it's an interesting situation where we actually just read two wavelengths, a green and a red, and by the configuration of the dyes, we can actually deconvolute all 4 bases using 2 channels. And this is shown to give very, very high quality data on all of our other systems, and so far everything that we've seen off of NovaSeq looks like it's also going to be extremely high quality off of the NovaSeq system. The thing about the NovaSeq system is that even though this flow cell is much larger and has many more spots in it, more wells, if you will, the scan time and the run time is actually decreased almost in half. So whereas the HiSeq X will do 16 genomes in about, that's 3 days, 3+ days, the NovaSeq system can do up to 48 genomes in less than 2 days. So that's a significant increase in throughput. In terms of ease of use, one thing that we do with all of our latest systems is that instead of using the way that we did it in HiSeq, where we use these trays with many different bottles that the users had to load in, sort of keep track of, make sure they were in the correct spot for all of these 20 different reagents. In the NovaSeq system, all these reagents are built into these cartridges, and so essentially it's just a matter of loading these 3 cartridges, loading your sample into one of the ports here in this cartridge, and then loading the flow cell, and then pressing go. So it's much, much easier to use. All of our consumables have RFID tracking on them so that the instrument knows exactly which instruments it's been used, or what consumables it has been using, and what experiment is being done, what even samples are on the flow cell can be sort of set up in your account before you start the run. The other key to this system is that we have really much better optics in this system than we ever were able to put into the HiSeq systems. And so we have sub-pixel geometric distortion, very nice nanometer scale laser-based focus tracking, and diffraction limited performance across the full field of view. And you might not be able to see this in this image, but this is one of the fiducials that we put on our flow cells so that you can see things. And if you look at the HiSeq X image here, if you look very carefully, it's actually quite distorted and blurry relative to the same sample or the same flow cell put onto the NovaSeq imaging system. And you can see that we get really nice resolution and really even, no distortion across either from side to side or top to bottom of the flow cell. So as I mentioned, this system is really fast in terms of its throughput. This is another way of looking at kind of the comparison I was doing at the beginning, sort of minutes per gigabase. This is actually gigabases per hour. And if you compare the NovaSeq 6000 system to the current state-of-the-art genome sequencer, the HiSeq X platform, you can see this is 4 to 5 times faster in terms of gigabases per hour, in terms of throughput per hour. So I just want to show a little bit of actual data from these instruments. And we do think that these instruments are going to do a lot of human genomes in the future, and so I wanted to show this data. This was data that we generated during our beta testing of this instrument internally, and this was using the S2 flow cell that is spec'd out at 1 terabase. You can see we're getting 1.26 terabases of data. The cluster passing filter numbers, the read 1 Q30 numbers, read 2 Q30 numbers are very good. The error rates, average error rates over the 2x150 base reads are very good. And actually, this data is available, this project in particular is available in our base space sequence hub, and I'll show you a little bit more about that in a second. But just to make a note, this was a TruSeq PCR-free 450 base insert library using the NA12878 standard sample and 2x150 with dual indexing. It was a 6 library sort of pooled that were sort of run together in the instrument. This is a little bit more detailed. We also ran the same set of libraries on HiSeq X, and so we're comparing the data in terms of where we downsampled them to very similar levels. We can look at the percent aligned. You can see they're very, very similar across the two platforms. Keep in mind that the HiSeq X system is essentially the state-of-the-art system today for doing human genome sequencing. The NovaSeq is comparing quite well in all the major overall metrics. And this just shows where you can go if you have an account in BaseSpace, or if you want to sign up for one, just go to BaseSpace and the Illumina's sequence cloud-based computing environment. And if you go there and go to the area called public data, you can find some NovaSeq data. I think actually now there's even more data up there than I'm showing here, but this particular human genome project that I was just talking about is actually in the cloud, and you can look at all the analysis we've done and also download the reads yourself and analyze them yourself if you're interested. One new announcement that we have just started talking about relative to NovaSeq in the last few weeks is a way to individually address all the lanes. When we launched the system in January, and when we started talking to customers about it in January and February, one of the biggest concerns was the fact that these flow cells are so huge and they can do so many different samples. So human genomes might not be that big of a challenge. It'd be basically 24 genomes per S4 flow cell. But if you start doing RNA-Seq or exomes or especially anything below that, you're going to want to sequence, maybe 100 samples or hundreds of samples per flow cell on with this kind of a throughput. So what we originally designed the system is that it has one loading port where one sample pool would be spread out across the whole flow cell. And what we've decided to enable is kind of a way to individually address the lanes on the NovaSeq flow cell. So here's a picture of what this will look like. It'll be essentially a device that you can preload your samples into the flow cell in an individual lane. So you can see that this S4 flow cell actually has 4 individual lanes across it, right? And so this will allow you to put 4 different pools of samples onto one S4 flow cell. And so just to do the math for you, if we have, say, up to 96 exomes or 96 RNA-Seq samples with unique dual indexes, that's another key advance that we're coming up with, is we're going to have new index adapters coming out over the summer as well that will have 24 and then eventually 96 unique dual indexes. These will allow you to take advantage of each lane, which will have about 750 gigabases of data. And so if you were doing, say, 96 exomes, that would give you 7.8 gigabases of data per exome. Or if you're doing RNA-Seq, that would give you 26 million reads per sample for 96 plex. So you could put like exomes in lane 1, RNA-Seq in lanes 2 and 3, and maybe some genomes in lane 4. That's what would give you the flexibility to sort of use all that sequencing power and be able to subdivide the flow cell into 4 addressable lanes. So now that we have the NovaSeq system, this is kind of our forward-looking view of our portfolio, where at the high end we have NovaSeq, which is can put out just enormous amounts of data, and at the low end we have our sort of baby sequencer called MiniSeq that actually puts out, it's a pretty impressive little instrument on its own. In fact, if you look back to the GA2 that I was talking about, this little baby sequencer that we have now is actually more powerful than the Genome Analyzer was 8 or 9 years ago. So this is our portfolio from benchtop to sort of floor standing instruments that can do panels all the way up to full genomes. And so the next part of the talk, I'm actually going to talk about applications that principally run on these smaller instruments, MiniSeq, MiSeq, and NextSeq. So these are targeted sequencing solutions for the oncology space. So I'm going to talk about, I picked just a handful of new and interesting cancer-related tools that we have developed to share with you some of the details today. So when you talk about using NGS in cancer, oftentimes people want to look at panels of cancer drivers, genes that are involved in cancer, and they want to be able to study, normal or germline tissue and look for changes in tumors and response and recurrence. And so we have a whole suite of targeted assays, actually, and you can see the sample types from blood to FFPE. This is FFPE blocks or blood, or bone marrow is also a commonly used sample type. In particular, I want to focus on these three panels: TruSight Tumor 15, TruSight Tumor 170, and the TruSight RNA Fusion. These are all different kits with different application areas and different workflows as well. So I want to introduce you to these three targeted panel assays. First one I want to talk about is TruSight RNA Fusion. This is an enrichment-based panel to study essentially gene fusions in RNA. So the challenge with using RNA-Seq to study known fusions is that in order to see a fusion, so here's a fusion between these two genes, ACAP9 and BRAF. If you look at sort of the coverage across the entire gene, you can see that the yellow half of the gene, which is ACAP9, there's lots of reads covering it, and the BRAF side over here, there's also lots of reads, but really the only reads that give you information about the sort of fusion itself are the reads that fall right along the junction here in the transcript. And so what we've developed is a way to targeted panels that we can sort of enrich for reads that are around those junction areas. And essentially what we do is we do what looks like two halves of a prep, where the first half is essentially a library preparation where we start with relatively small amounts of RNA, and the RNA can be FFPE as well. And we make a stranded total RNA protocol or total RNA library. Then we take that library here and put it through an enrichment method where we used oligos that are targeted to the genes of interest to capture them and essentially wash away all the genes that we're not that interested in a way do a focused cancer study on just the genes of interest. And so, for instance, in this fusion panel, we're targeting 507 of the roughly 23,000 genes in the human genome. So these are genes that we know are involved in fusion events in cancer, and so this is a way to kind of study those genes directly. This assay actually gives very robust performance even if the RNA is of very poor quality. And so a couple of years ago, the scientists at Illumina developed this new metric that we call DV200 that allows us to essentially quantify how much RNA is worthy of making a library, essentially. And what we found is that we can sort of dose in the recommended input amount based upon whether it's relatively high quality RNA, it could still be FFP, but it's relatively high quality, where it's greater than 70% DV200, to really poor quality, where you have to put in even more than 100 nanograms. We usually don't recommend it, but actually some users actually do take these really degraded samples and put in 100 or 200 nanograms to see what they can get. You can see that these samples are truly degraded here based upon the bioanalyzer traces here, or the fragment analyzer traces that we're showing here. And so the way this works is, picture you have a fusion event between two genes, and you want to capture that. Essentially what we have done is designed probes against sometimes only one of the genes would be in our pool, and the other gene might not be in the pool, which is shown here. So we have probes, 80-mer capture probes shown here, that are targeting these green fragments. What is interesting and what makes this assay really a nice discovery tool is that even if the sort of gene over here, gene number 2, is not targeted, we will still capture fragments, RNA fragments that span that junction. Essentially, it's a novel exon-exon junction in these fusion transcripts, where you can pull this down, and by knowing what the green gene is, you can sequence across that RNA fragment and essentially discover novel partners, even if you only know one of the partners in the fusion event. And so, for those of you who are interested in this or study this, you know that in many important fusion events, like those involving ALK, for instance, ALK tends to form fusions with many different partners. We can find those fusions using this assay, even if we don't know what gene 2 is, and that's what makes this really a powerful approach. This is some of the data where we have used this and shown that we can detect fusions in FFPE samples. I didn't mention this before, but this assay is generally run on 8 samples on a one mini-seq or MiSeq run. So that's what we're showing here. These are 8 different samples that have been multiplexed together on one MiSeq run. And you can see that we are detecting, even in poorly samples that have very high levels of degradation or low DV200s, we are finding sort of the expected fusions that we are looking for. We've developed some really nice software for analyzing this. We have a local run manager, LRM fusion module, that if you run this on mini-seq or MiSeq, you can just analyze the data directly and get a report like this, where we will report out high confidence fusion calls or low confidence fusion calls, or if you're interested, you can see all the fusions that we looked for but didn't find in the data, so recurrent fusions that are not called in your particular data. So this is a really nice report. It tells you the disease associated with these fusions, the actual gene coordinates involved, the number of reads, and sort of the confidence levels that we have in the call. This table kind of gives you a little bit more details. This information is available on our website, so I'm not going to dwell on it. I do want to just say that we have a companion panel that's quite a bit larger, 1,385 genes. It includes all of the 500 in the fusion. It also has more genes that are involved in alternative splicing and gene expression sort of markers in cancer. Very similar specifications. You can see there's obviously a lot more regions that are targeted in this larger panel. We recommend 2x76 base pair reads and about 3 million reads to collect for these samples. So like I said, if you're interested in targeting RNA and especially fusions, these TruSight RNA pan-cancer, TruSight RNA fusion are really nice new products that we've put out in the last 6 months or so to do that, and there's much more information available on our website. The next sort of assay I want to talk about is called TruSight Tumor 15. This is a completely different approach. This is basically a DNA amplicon-based focused panel, and so this is more like a PCR amplicon assay. It's much smaller in terms of the targeting and much more focused on a multi-gene cancer panel to cover these 7 types of diseases, and will give you readout on important mutations that are involved in some of these diseases. You can see many of these are some of the most famous and highly studied cancer genes in medicine today. And this panel is designed to give you a really focused, a really high accuracy readout and very sensitive readout of all these genes. This is the sort of content and the regions that are targeted, and specifically the mutations that are targeted for this. This just shows a little bit about how the assay works. It's really a pretty straightforward PCR assay where we leave a little bit of an overhang on our starting PCR primers that target the gene-specific regions, and then we sort of do a second cycle of PCR where we include all the what we call the I5 and I7 regions to create the adapter, so that this construct here at the bottom can be sequenced in our system. So we start with initial primers, extension primers, so that you end up with this construct at the end of the process that can be loaded onto any of our sequencers. This assay is very sensitive. It has about 250 amplicons in the assay. only requires 20 nanograms total. We actually split it into two 10 nanogram reactions to do two separate pools. It's 8 sample throughput using the MiniSeq or the MiSeq v3, and it can call variant frequencies robustly down to 5%. Some folks even push it below that. And when you're using this assay under these conditions, 93.5% of the bases that we are targeting are covered at greater than 500x coverage. So you can get very deep coverage on this, which helps you to have those low variant frequency calls. This shows once again some of the data using this assay in an FFPE sample, and you can see that this shows the quality of the FFPE that was used, and you can see that very low quality FFPE, we still get extremely nice coverage of the percentage of bases that are at 500x or higher, and then sort of the mean amplicon coverage you can see is tens of thousands for most of these samples. And so this is what allows you to get, extremely rare variant detection. If you're going to call below 5% or even at 5%, and with good statistics, you want to have thousands of fold coverage so that you get mini reads that are sort of representing the mutant allele. So yeah, this to remind me to say that these actually, if you're used to measuring DNA delta CQ using a PCR assay to study the degradation of the DNA, these samples here are as low as delta CQ of 4 to 6, which is fairly degraded FFPE type samples. This assay is also very accurate. We use some standard samples to look at sort of expected variance of these samples versus the tested variance, and you can see we get almost a perfect straight line between variants that are all the way down to 5% all the way up to above 50%. And so this is all done from FFPE. This is part of the sort of validation work that we do when we launch an assay like this. This is extremely high performing assay, like I say, all the way down to 2 10 nanogram pools of FFPE material to get these results. And finally, the last assay I want to talk about is called TruSight Tumor 170. It's an enrichment assay back to so the first assay was a sort of an RNA enrichment assay. Second assay was an amplicon assay. This assay is a DNA based enrichment assay that also has a little trick where we could do RNA at the same time. And so this is much more content than the previous assay, up to 170 different genes, and it's essentially sort of a TruSeq library plus hybrid capture. We also call minor allele frequencies all the way down to 5%. It requires just 40 nanograms of DNA and RNA. The neat thing about this assay is it can actually prep both the DNA and RNA from your samples. We have very nice informatics suites around this. We have automation protocols around this prep as well. And it says here coming towards the end of 2017 for custom solutions. We have launched this assay already this year. So this just shows some of the mutations that you can detect with TST170. And in DNA, you can detect single nucleotide variants or insertions and deletions or amplification products or even phased mutations where you have several mutations all in the same area, but on one strand. And in the RNA world, we can detect gene fusions like I've talked about already, and then also splice variants. This panel is a much larger panel than the TST115 panel. It includes many more genes and a couple of different diseases that are not necessarily covered in the TST15. And remember, we're targeting both DNA and RNA with this panel, and so some of these assays are targeting known RNA fusions, which are really important for certain diseases. This is kind of the types of, this is the gene list showing the types of variants that we're looking for in the sort of blue bubble here, we are looking for small variants, single nucleotide variants. In this purple bubble, we're looking for RNA fusions and splice variants, and in this green bubble, we're looking for amplifications of gene amplifications. You can see there's a lot of overlap. Some of these genes here in the middle, we actually are kind of looking for all three things potentially happening in those genes. So the assay, like I said, is unusual in that it will start with either 40 nanograms of FFPE DNA or 40 nanograms of FFPE RNA, and go through a library process where you'll end up with a library of DNA and RNA converted to cDNA, obviously. And then at this point, these are very similar in terms of their ability to work in an enrichment reaction. So then we carry those library products on into a capture-based enrichment where we use probes to pull out all the information around those genes, and then we can analyze it on the sequencing, and we can tell based upon the types of sequences we get whether that came from DNA or RNA, and we can call all the various mutations that we were talking about on the previous slide. This just shows the performance of TST-170, that we can get really nice coverage even with very low quality. So this is along the x-axis here is delta CQ, where the higher you are here, you are starting to get really highly degraded samples with delta CQ of 6, 7, 8. On this axis is the number of exons that we target at 250x, and you can see that it's pretty much in the high 90s all the way out to delta CQ of 6 or so. So that's where we spec this assay out. And you can see some of the other sort of specs here at the bottom of the slide. But the point is we get really nice results even with highly degraded DNA. And this is some of the validation data that we've generated using Horizon samples. These are very nice standard samples that have variants across this set at known levels, which are shown in green here, and then what you can see is what the assay reported in blue, and you can see there's a very good concordance across all of these variants between what we expected to see and what we actually measured or quantified. This is showing that we can also detect amplifications between different samples and even look at variants in RNA, structural variants in terms of RNA fusions using RNA that has very low quality. Once again, we're using this DV200 metric. And so we're able to pick up copy number variations for different regions that are very clear using this assay. So this just shows some of the actually beta testing data that we did during the validation of this assay. And so these are 3 of the groups that worked with us on this, and you can see that they detected, for instance, these variants here. We know these, we expect to see these in samples like the Horizon samples, and they have an expected variant frequency here, and you can see that all 3 of the users essentially got excellent results and very nice concordance, even quantitative concordance with the expected variations across all the sites. So this is a really robust, really accurate and quantitative assay. As I said, we have the RNA aspect to it where we can detect fusions, and that's what we're showing here. These are expected variants that were all detected easily at all 3 of the sites using the TST-170 workflow. And then this is just showing that the measurements that we make also have very high correlation with whole exome sequencing. And so the variant calls that are called here are very similar to what you would get with whole exome sequencing, which is another type of enrichment assay. And so this makes a lot of sense. And finally, this is information that, once again, more detailed information on the TruSight Tumor 15 kit that we talked about and the TruSight Tumor 170. Again, this detailed information is available at our website, and if you're interested at all in sort of targeted cancer solutions, I think these three kits should be of interest to you. So in conclusion, I've tried to introduce our new system, which we think is going to sort of really push the boundaries of what people can do with next generation sequencing. And then also three, I think, of the more interesting new applications that we have developed to study cancer and translational research. In particular, the TruSight Tumor 15 amplicon assay, the TruSight Tumor 170 enrichment assay that can enrich both DNA and RNA, and then also the RNA fusion assay, which is really a much more open-ended discovery tool for studying RNA fusions. So that brings me to the end of the presentation. I'll just remind you of our portfolio again, from the mini-seq at the low end all the way up to the new NovaSeq 6000 at the very, very high end. Really, we have sort of a variety of sequencers for a variety of applications, from human genome sequencing and lots of RNA-Seq and exomes up here, down to some of these targeted panels and targeted solutions that we've been talking about that run more on the NextSeq, MiSeq, and mini-seqs. So with that, I'd like to thank you for your attention, and once again, I'd like to thank LabRoots for inviting me.
Thank you, Dr. Schroth, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many questions as we have time for. So let's get started. Our first question is, can I run different sequencing methods in different lanes on NovaSeq using the lane loader device?
So that's a good question. So that lane loader device is designed to allow you to address any pool of samples into so let's say in the S4 configuration, there's four lanes that you can address. So yes, you could put exomes. Let's say you want to do exomes and RNA-Seq and microbial genomes and human genomes. You could pool those all together and do all the exomes in lane 1 and all the RNA-Seq in lane 2 and the microbial genomes in lane 3 and a handful of human genomes in lane 4. So yes, that's possible with that lane loader device. That's really one of the main motivations for us putting that into development.
Can the fusion kit detect novel RNA fusions?
Right, so the TruSight fusion kit that I talked about, it targets 507 genes, and as I tried to show in that diagram, it can also target a wider variety of genes if, for instance, the one half of the gene fusion is something that we're probing for, then we should be able to pull down fragments from the other side and capture essentially novel transcripts or novel fusion partners. And so yes, that's kind of one of the most important features of that assay, is that it can easily detect all the genes that are involved within the 500 set, sort of fusions between any of those 500 genes. But you also have a very good chance of capturing a fusion event between any other gene in the human genome and any of those 500 genes.
Great. How well do the cancer panels work on FFPE samples?
So all of the cancer panels that I talked about today, the TST15 Amplicon panel, the TruSight RNA fusion panel, and the TST170 cancer panel, they all work really well on FFPE. As I mentioned, if you are interested and you go to our website, illumina.com, and look under cancer, you can find a lot more specifications about those kits. But I believe they all work down in the same range of 20 to 40 nanograms of input of either RNA or DNA and that input applies to FFPE as well. And so yes, all three of those kits work really well with FFPE.
Okay, for the next question we have, does index hopping occur on NovaSeq?
So index hopping actually, to some degree occurs across all of our platforms, and so yes, it does occur on NovaSeq, and we have. I didn't have time to get into that today. But we have been developing a lot of new products and to sort of address the index hopping issue. Just to go through the information on that, we actually have a lot of information about that on our website as well. We have a whole page dedicated to index hopping if you're curious about that. But I'll just kind of cover the three main recommendations we have for index hopping is to first clean up your libraries very well and remove any free index adapters and primers. We find that is probably the biggest driver in causing index hopping. And the other thing that we strongly recommend is to use dual indexing and unique dual indexing at that, where you have actually unique pairs of indexes on either side of the fragment of interest, so that if you're using, say, a 16-plex library pool, you have a set of indexes on each side that is unique for all 16 fragments that are in there, 16 libraries that are in the pool. That allows you to do dual indexing, and it allows you to explicitly identify any index hopping and eliminate it from the data so that it is completely not part of your analysis. So what we've found is that index hopping does happen at very low levels, much less than 0.5% generally for almost all applications, and that using these two sort of preventative methods where you actually basically make sure you're having very clean libraries and then design your experiment in a way that you can use unique dual indexes, it's really not a concern for all applications these days. And so NovaSeq will be able to you'll be able to perform these very highly multiplexed experiments on NovaSeq without having to worry about index hopping.
Okay, for the next question we have, can I use custom primers on the NovaSeq system?
So that's actually a pretty easy question. The short answer is yes. The cartridges that we use that you port your sample in actually has a couple of open ports that you can put in custom primers, and you can easily program in the software for it to use those primers for various priming events. So if you have sample prep processes that you're used to using that require custom primers, or if you're developing a new assay, let's say, that you want to try out custom primers, that is possible to do on the NovaSeq system, just like on many of our other platforms.
Okay, it looks like we have time for one more question. What is the maximum read length for a sequencing run?
So I assume that question is about the NovaSeq system. The NovaSeq system actually allows up to 2 by 150, so 150 base pair paired-in reads. If you remember, the kit configurations that we are going to offer with this system are 100 cycle, 200 cycle, and 300 cycle kits for at least some of the flow cells, and there's a variety of configurations. But essentially, with the 100 cycle kits, you could do actually single-ended 100 reads, or you could do paired-in 50, or you could do a 75 times a 25. They're actually quite flexible. With the 300 cycle kits, you can go up to 150, so you could do 2 by 150. With the 200 cycle kits, you could do 2 by 100, or you could actually do a 150 and a 50. So there's a lot of flexibility, but on the NovaSeq system, as currently configured, the maximum read length in either read 1 or read 2 will be 150 base pairs.
That was great. I would like to once again thank Dr. Schroth for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. And before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August of 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Goodbye.